So how are those two subunits going to combine to form the ribosome? The condition required is for assembly is magnesium ion concentration. If the magnesium ion concentration is 0.001 molar magnesium ion concentration, then the two subunits assemble and we get a ribosomal structure which is visible to us. That is, and this is for our understanding, we draw the structure, but we have drawn the subunits separately. So this is the larger or large subunit, and this is the small or smaller subunit. So what is required for assembly is magnesium ion concentration this much. If it is less than this, then these two subunits will separate. If the concentration is more than this, if it is more than 0.001 molar, then what is going to happen? Then two ribosomes, they assemble together. Here we have seen a single one. In this condition, what will happen is, this is one ribosome assembled and this is another ribosome, and they come together. This is known as a dimer. So what condition is required for formation of a dimer is higher. So it has to be more than 0.001 molar magnesium ion concentration. So we are talking about magnesium ion. If it is more than 0.001, dimer. If it is 0.001, simple assembly. If it is less than that, those two subunits are going to separate. So magnesium ion concentration plays an important role in the structure formation. Sometimes when multiple polypeptide chains are to be formed, then many ribosomes join together to form a chain. Those chains would look like this. Such a structure or a chain of ribosome is known as polysome or polyribosome. This is formed only when multiple polypeptides are to be synthesized. So we have seen how assembly takes place. Then what happens if the concentration of magnesium ion is little more? We will get a dimer, and a polysome can also be formed, or a polyribosome can also be formed. Now what exactly is the role of this ribosome? Ribosomes help in protein synthesis. So their main function is protein synthesis. Let us try to understand how this ribosome synthesizes protein, then how does it come to ER. We have just seen in the endomembrane system that endoplasmic reticulum takes and transports these proteins. Then those are sent to the Golgi body, lysosomes are formed. So we are trying to see how these things are happening. This is actually a schematic diagram in which we are trying to see how these connections are there and how what is produced where and transported. This is endoplasmic reticulum. Suppose this is a section of the cisterna of ER. Cisterna of endoplasmic reticulum. Attached to this is a ribosome. We said that which part or which subunit attaches to the endoplasmic reticulum is the larger subunit. So this is the larger subunit. Again, this is a schematic thing, so this larger subunit does not exactly look like the subunit. We are drawing it only to understand how the system works. And this is the smaller subunit. Let us put the labels here so that we are able to correlate. This is larger subunit, and this one is smaller subunit. Larger subunit has some part in the middle. This is a channel in which polypeptide chain is synthesized. So this structure which we have drawn here is a channel for polypeptide. So the protein or polypeptide molecule which will be synthesized will come here, and see from this opening it will come into endoplasmic reticulum. In the smaller subunit we are seeing a depression here. This is the place where mRNA is going to get attached. So say this is the mRNA molecule attached to this depression in the smaller subunit. So this structure is mRNA. With the help of mRNA, these ribosomes, they help in the synthesis of polypeptide. This polypeptide which is synthesized would move into endoplasmic reticulum. So this area through which these polypeptides are coming in are the channels, and we have not drawn this. Here we can draw a small thing which is actually a protein with which the larger subunit is attached to the endoplasmic reticulum. So this small line which we have shown here is ribofourin. So now how these two subunits helping? First of all, when protein is to be synthesized, the two units should join. This is known as assembly, and we get the complete ribosome. The smaller subunit has a depression or a site for mRNA attachment. The larger subunit has a channel from where the protein polypeptide will be pushed into the endoplasmic reticulum. When this assembly takes place, it binds to the ER with the help of a protein which is on the ER membrane called ribofourin. The information which is on mRNA is translated into this polypeptide. What is ER going to do? ER has taken this protein, and from this cisterna a vesicle will be pinched off, and this protein can be sent wherever it is required. If it is to go to Golgi body, it will go to Golgi. If it is just required in the plasma membrane, it will go there. So these are the vesicles which are going to carry this polypeptide which is synthesized by the ribosomes. Once this complete process is over, then all three things separate. That means ER and ribosomes and the two subunits of ribosome. So polypeptide goes wherever it is required, ribosome detaches from ER, and what is required for detachment is lower magnesium ion concentration, less than 0.001 molar. Once the ribosomes separate, the two subunits also separate. So the function of ribosome is protein synthesis. So the protein synthesis is a different chapter, maybe later on we'll do that chapter also, but I hope this complete structure has explained how ribosomes are helping in protein synthesis and how ER packages it. So ribosome, important things is, main thing is this concentration, more than this, less than this, what all changes are seen because of these concentration, and we have talked about some scientists and their discoveries into it. So this completes our ribosome part. Let us take another organelle now, that is vacuole. Vacuoles are found in plant as well as animal cells. In prokaryotic cells, we don't have vacuoles. So they are found in all eukaryotic cells like plant and animals. Again, the exceptions are mature mammalian RBCs. Only thing is, in plant, the vacuole is large and the number is normally 1, but it could be 1 to few, very few. And in animal cells, they are small and the number is normally more. So there are many vacuoles which are present in this. The shape of a vacuole is normally spherical or oval. So it's a simple circular membranous or membrane bound structure. The membrane of a vacuole is known as tonoplast. It's a typical plasma membrane type of structure known as tonoplast. The things which are filled inside are different, but we are talking about a general vacuole. So most of the time when term only vacuole is used, it has sap inside. And this sap is normally acidic. Normally acidic in pH. What is stored inside the vacuole would give us various types of vacuoles. So types are basically on the basis of what is inside it. First is the same, which is known as sap vacuole, or whenever we use the term only vacuole, then also we are talking about sap vacuole. So only vacuole or sap vacuole, one and the same thing. It has sap. These are predominantly found in plants. The second type is known as contractile vacuoles. Contractile vacuoles, they are found in freshwater organisms, organisms like amoeba, for example. Now in freshwater organisms, normally what happens is, if say this is amoeba and it is in fresh water, because of the difference in the concentration, again this is going to be hypotonic as compared to the cytoplasmic content of the cell, which is hypertonic. So endoosmosis will take place. Water will move in from the surrounding. So there is a risk that this cell might swell and ultimately rupture. So these contractile vacuoles keep collecting this water. So they collect water and by exocytosis they expel this water outside. So basically they are helping in removal of all that water which is passively coming into the cell. So this regulation is known as osmoregulation. And that is why it is said that these contractile vacuoles work like higher animals' kidneys. Our kidneys also help in osmoregulation, and so are these contractile vacuoles. One more thing which happens with the help of these contractile vacuoles, when it is collecting water and removing it, some waste material or excretory materials get also eliminated with it. So it is also helping in excretion. Both the functions similar to the kidneys, and that is why it is considered as the same kind of organelle which is performing the function of kidneys. So this is contractile vacuoles. If you remember the structure of paramecium, the slipper animalcule, there is a gullet here, and we draw two contractile vacuoles here. Normally these contractile vacuoles are made up of a central vacuole and many radiating vacuoles. So it normally looks like a flower, and there are two, one on each end. So These are contractile vacuoles which are present in paramecium, performing the same function. So it is a characteristic feature of freshwater organisms where passively endosmosis is resulting into more and more water coming in, and they have to get rid of that extra water. The next type of vacuole after sap and contractile are called gas vacuoles. As the name tells us, these vacuoles are nothing but balloon-like structures. Same plasma membrane which is known as tonoplast, and inside is gas. So these are actually bubble-like things but membrane bound. These are found in structures which are normally in water, so it can provide buoyancy. So this balloon-like things which are inside, they help the organism or that structure to float. Sometimes other than sap or these functions, it also has some pigments. So these are called pigment vacuoles. The pigment vacuoles would contain pigment. Normally the pigments which are found are anthocyanin, or xanthophylls. So depending upon that pigment, that vacuole would even impart color to that particular part. One more type of vacuole have been reported which contain excretory material. So those are known as excretory vacuoles. So they just keep on accumulating the excretory material or toxic substances from the cell and ultimately may release it outside by exocytosis. So this is also for removal of excretory material. So these vacuoles are very simple structures, simple membrane bound, found in all eukaryotic cells, and depending upon what they have inside them, there would be different types, and depending upon this there would be functions. Like for example sap. Sap has minerals, ions, sugar. So basically the substances which are to be stored inside. So this sap vacuole also helps in providing turgidity. So now depending upon these types, let us sum up the functions. What is the sap vacuole doing? It is storing minerals, ions, sugar and various substances inside the cell. If you remember the plant cell diagram, there was a big vacuole, that was the sap vacuole. Now cell enlargement is also because of this sap vacuole. As it becomes bigger, the cell also goes bigger. Plus more and more water into the sap will make the cell turgid. Turgidity of cell is required for uncoiling of leaves or petals. Normally in a bud the petals are crumpled. They open up because the upper cells, they become turgid, they become larger. That is also with the help of these sap vacuoles. Contractile vacuoles remove all extra water plus some excretory material gets also removed. Gas vacuoles provide buoyancy. Pigment vacuoles, they store pigment, so provide that particular color to the part of the plant. Excretory, they are going to help in removal of all the excretory material. So vacuole, a simple membrane bound structure and very basic things about it.